Welcome to Fertility Frontiers.
I should be here.
I had the feeling that most of the people that is going to have access to this episode would know who you are. But in case there's people that don't know, today we have Dr. Jacques Cohen, who has co-developed multiple groundbreaking embryological methods and devices. You have over 300 publications. You own over a dozen patents, and you hold a number of advisory roles in multiple biotech startups. You're also the CSO at Conceivable, and that is my main focus for the episode today. So welcome, Dr. Cohen.
Thank you.
I wanted to try something to get to know you a little bit more, not only as a researcher, but just as an individual. And I have some rapid fire questions. So your aim is to answer in 1 or 2 words.
Okay.
Okay? Let's go. Early riser or night thinker?
Both. I dream all day. I dream at night. I dream an hour awake. Steven Spielberg.
Yes, the definition of an entrepreneur, that's for sure. Okay, coffee or tea?
Oh, yeah, sorry. Both too. Both as well.
Okay. If you weren't in relative medicine, what would you be doing?
I'd probably be very sad because right now I would know what I've been missing. I just love this field, but I would have probably be an artist, most likely, a photographer. Used to be a sculptor, so it'd be something like that. I'd be very poor.
Awesome. What is the most surprising thing about embryology that most people don't know?
I think most people don't realize the intensity of it. It takes over your life. It's day and night. And it's a job that and frankly, in my case, I was an embryologist only for 20, 22 years. It could have been 40 or 50 years, but I started being an experimental embryologist in 1976, and I could only do 20, 22 years of embryology. I thought it was way too intense. And it may have been the time, but I don't think it has gotten any better.
Really good answer.
Yeah.
It is. So what is the most inspiring scientific book or paper you've read?
Oh, it's a lot. I'm interested in science in general, and I do read a couple of journals in general science. And so it's difficult. I can't really point out. It's always the last one or the second to last one, but I can't point out.
That's a fair answer.
Yeah.
Which discovery in IVF history fascinates you the most?
I think that every time when there's a fusion or a convergence of two completely different fields. And so the field of engineering is very broad. If you look at genetic engineering, when PCR was developed in the mid-80s, it took only for Handyside and others, Alan Handyside, to apply that to single-cell biopsy. It was done at the time at the 8-cell stage. And I think that was a tremendous step forward to be able to, in a living organism, be able to take a single cell out and study its genetic profile. That's really changed our field. And the same happened with cryobiologists. Usually they were biophysicists or physicists and engineers, where they developed just years before that, a decade or so before that, cryopreservation of oocytes and embryos, mostly embryos. I think that was another one of those occasions. And you see that happen from time to time. So it's usually some form of engineering that fuses or converges with IVF. And those are the good moments. Don't shy away when an engineer knocks on your door. Don't say, no, I don't have time or anything like that. It's usually a 100%. Good occasion to proceed and consider what they have to offer.
What is a scientist or researcher that you deeply admire?
I think Ray Kurzweil. He's also an engineer. Ray Kurzweil, you may know him from the Kurzweil synthesizer. I think he was the first to develop a digital way of Braille, so reading for blind people. It goes back to when he was in his teens. And I think over the years, he's produced so many breakthroughs that I think that's probably one of my heroes. But so is Richard Dawkins, because he fits well with my look of life, although not always admired in his own environment. But yeah, I think he fits that profile as well. There are quite a few that are not with us anymore. So I mentioned 2 that are still with us. Of course, Einstein, Galileo, Edwards, and I think that's all in the same breath. Okay, I think Bob Edwards fits that. So probably in our field, of course, having worked with him, that's probably the person I've been inspired by the most. Yeah.
What is the best advice you ever received in your career?
Never give up. That's probably one of them. And that came from a colleague when I was doing my PhD. When you do your PhD, you have a very strict, and also a master's degree in research, you have a very strict timeline, but you need to give yourself time to solve things. So yeah, never give. This is, of course, an advice that a lot of people get. But yeah, you have to try again. Do something different. Don't go by what the paper which you base this on, materials and methods of somebody else, is based on. They usually leave something out. I'm not saying on purpose, but because it's so obvious to them. They perceive this I can't write up because that is obvious. And that happens all the time. And so when you repeat that, in my early years of science, you need to understand there's no internet. There was no internet. There was no social media. There's no communication other than the people you work with. So copying a paper and trying to send up the methodology, yeah, don't give up. Try to do something different or give them a call. We did have telephones, so we could call them and maybe get them on the line. So that worked. That you need to be outgoing. You can't be reclusive and do this all by yourself.
Fully agree. Okay, so in your opinion, what is the most promising startup in the IVF field at the time?
I think there are quite a few of them. You probably want to hear from me that's Conceivable Life Sciences. Certainly that is one of the most exciting ones in my lifetime, which stands all of IVF, the development of IVF. Right now, it's all those efforts involved in automation and also AI that is very inspiring. There have never been as many startup companies and academic efforts associated with AI, associated with a new technology, in this case AI, in our field as ever before. It's over 30 companies, and it's at least 20 or so academic centers on top of that kind of interest I've never seen before. You can call it hype, and in a way it is, but usually hype, when it has so much attention, it usually settles down and becomes real, becomes realistic. That can take years, but it usually at some point happens. So seeing that there are many companies in automation, Fertility, or Auto IVF, Overture, scientists that have worked on this in the early days, Gary Smith, Professor Yusan from Toronto University, who I think is the founder really of this field of automation and also AI. I think I admire all of that. I can't really lift out one company, but I do think I'm associated with one of the most exciting companies in conceivable life science. Yes.
We have mentioned a couple of companies and startups that they are working on automation in the IVF lab. So if you want, I would like to move into speaking a little bit about Conceivable and what they're doing. But before we dive into the technology that you guys are developing, could you give me an explanation of the different approaches to automatize the IVF lab?
Yeah, that's a very good question. And thank you for asking that. There are really two, but there could be also combinations. But there are two main ones. They both started around the year 2000. Gary Smith, in our field, was really the founder of the microfluidic miniaturization approach. We owe him a lot for that. I think most of the efforts in engineering and embryology in automation have followed that line of thought. And we have some relatively simple microfluidic devices already available to us, mostly in the arena of sperm preparation. And I think that has been a good development or well. And then the other way, it does not look necessarily at miniaturizing everything on a microscopic or on a Petri dish level, on a slide level, but look more at what do embryologists do and how can we imitate that or use that to our benefit in automation. And that's the approach that Conceivable follows. I think probably the first group to do it, but it's based on the pioneering work of Professor Yusan from Toronto University. I already mentioned him before. His first publication is also in our field, is also in 2000. So we're looking already at 25 years of research and trying to define and see if it's possible in certain aspects of what embryologists do to define microfluidics, miniaturization, and imitating larger robotic steps. So those are the main drivers. I think the last one, started by you, son, I think is the one that has had the least attention That's what Conceivable focuses on. By no means do we exclude microfluidics as a solution. We just feel that there are some advantages to doing general robotics. General robotics has been done or is being done in cell tissue culture laboratories for different purposes, a lot of to do with pharmacology. And so those tools have been made and are available in industry, not in IVF industry, but in the cell biological arena. And so we shop there. That's where we get our tools, and then we adapt them to what embryologists do. All the pipetting, automated pipetting has been going on for 20 years. You can buy an automated fluid system for pipetting for general pathology and diagnostic laboratories 20 years ago. And Hamilton Company, I think, started that. That's different from Hamilton Thorne. Hamilton Company started that. But that's mostly vertical. So now how do you add vertical or other degrees? How do you add that to it? That's very typical to IVF. So that's a difference. So we do have to build that from the ground up and can't buy a Hamilton machine or a derivative of it and say, this is going to work in the lab. Some of it could, but most of it would not. Fluids control in our field is very detailed and specific to our field. It's not in any other field. So that is the difference.
Where are you building this fully automated IVF lab?
So Conceivable was founded by Joshua Abram and Alan Murray, and another co-founder is Alejandro Chavez Badiola. And because of Alejandro and my earlier collaboration with him in a company called IVF 2.0, which still exists and develops AI products for the IVF laboratory, because of that, I noticed that this was in Guadalajara, where he was based. He also had a clinic in Mexico City. I noticed there were so many excellent engineers. There's a huge engineering university in Guadalajara. It's about 60,000. It's massive and so much talent. Even if of 60,000 students, only 10% are talented, you can imagine the sheer volume of it. And so that was that IVF gave us that confidence to go to Mexico City. And I have to give credit to Joshua and Alan believing in that and seeing there was a future there. So yeah, our main research engineering team is based in Guadalajara. And the first line of the clinical prototype line that we call Aura was built in Guadalajara, but then set up in the IVF clinic called Hope in Mexico City. So we work with engineers in Mexico City. We also have associations in Spain, in Embryo Tools in Barcelona, Nuno Borges Laboratory. And also we have engineers in Britain working on various aspects of the development as well. So it is very international.
And so that laboratory that you have developed in Mexico, what are the steps through the IVF process that it can done automatically without any kind of assistance by a human?
You're talking about autonomy. I don't think anybody has gotten to the autonomous level yet. However, we're very close, depending on the steps that you're looking at. So sperm prep, we prefer on most samples to do what we call horizontal swim out. It's actually a method that's been published indirectly or directly over the years. We didn't invent horizontal swim out. It probably goes back decades, but it was perfected by Spanish researchers pretty recently. And you basically have little droplets of medium and you add sperm to the first droplet and then swim through narrow channels that you pipette and leave the whole thing under mineral oil. And you go from droplet to droplet and then end up in a final droplet. And you can make different configurations. So we have a configuration we call the spiral, where we start at different little droplets with semen added, and they end up in the central droplet. They could even end up in the PVP droplet. And so then you're ready. You don't need to do anything. It's just a simple step. So that we have, I would think that's autonomous, robotically autonomously derived. It's just a start, basically you switch it on. But does it work all the time? Most of the time it does, but it is still a clinical prototype. It still needs fine-tuning and optimization. Typical processes of engineering that can take a while. And the other one is dish manufacturing. Also, that is very close to autonomous. Autonomous, also that needs considerable optimization still, but we're very close there. And then the ICSI process, we operate with 23 major steps in ICSI, but you can subdivide them, and then there are over 100 steps. If you go into the coding level, or even more than 100 steps, But if you look at the original 23 steps, bring the tools and the egg into focus. It's one step. Those steps are, in the majority of cases, autonomously derived. You just click on a screen and say, bring the whole thing into focus. And it does it 9 out of 10 times or 98 out of 100 times. So that is autonomy. And then once you achieve that at a high frequency for those 23 steps, what you then need to do is stitch those steps together. You basically say, you finish one, now you can start the next one. And when you do that, you can you click on ICSI, you click on that, and then it will do ICSI on that egg or that group of eggs. Have we achieved that? Yes, once or twice. But now we have to do it at a level where you can say we can achieve that, let's say 90% of the time. And then if that doesn't work, you still need digital control. So it's not just those 23 steps that you can click on. You also need to be able digitally, as an embryologist watching this or doing this, you need to be able to control magnification. There needs to be an automatic turret system of some kind. Or there needs to be a stage that's moving and you control it as an embryologist and you have those. It basically is like doing a complicated game. It's like a gaming framework. And that's where a lot of it comes from. And so you have to have controls of suction. You have to have controls of the micromanipulators. And once you do all that and a command doesn't work, then you take over digitally. Not that easy, by the way. It's not something you can do just from scratch the way we do it now. It's pretty difficult, actually, because you go from something where you have a feeling. It's all feeling, right? When you do micromanipulation, you can have actually have feedback that you don't have on the digital control. So we really want to go to autonomy and do minimal digital control because it's not that simple. It's very flat. It's very indirect. Sometimes frustrating.
You can basically, even if it's not fully automated, but you can do all of the steps from finding the eggs in the dish to doing the fertilization to growing the embryo and even freezing the embryo. Is that correct?
That is correct. Yeah, that's what we're trying to achieve. But there are still some elements that we need to add on to that. I love to do IVF, and that will be a relatively easy addition. We need to achieve that. And I think we can actually go back to IVF and make it work. It's a method we have neglected as a field. It really is unfair to the technology. And I think there's a major advantage not penetrating the membrane. The fact that we get away with ICSI is actually miraculous. We should not be routinely penetrating the membrane. So you can see that from the development of Dr. Hatakeyama from Japan and his group, recently published in Fertility, but first publications are about 5 years ago, where they go to the zona pellucida with a needle. Actually, before that, pick up a sperm on a zona pellucida through IVF. Take the one that immobilizes or is less mobile, take that off because it means it's overreacted. And you pick up that sperm, not that easy. You pick up that sperm in a typical ICSI-like needle, you go through the zona pellucida at 12 o'clock because you don't want to penetrate the membrane. And then you hold that sperm for about 20, 30 seconds on the membrane. It is a touchy technique and probably takes some time to really get good at it. The advantages are amazing. No lysis. There's no degeneration of the egg. And the fertilization rate is either as high as the Vienna KPI or it's higher. It is frustrating to see eggs die from ICSI, and they do. Let's not deny it. Not much literature about it, but we know that eggs die. The eggs also die from denudation a lot in some clinics, and by some starting embryologists, trainee embryologists, have a very high rate of degeneration, both from denudation, from doing ICSI. It's a completely natural thing. But even if you're very good at it, eggs are still dying. Even if you're very good at clinical ICSI, at standard clinical ICSI. So I think this sperm egg binding fusion method, which is surprisingly rapid, nobody thought until the doctors in Japan started doing this, nobody thought that it would only take 20, 30 seconds to achieve binding between the membrane and the sperm. But you have to expose them to the zona pellucida to do that activity. So it's a very natural process. You're actually going back to IVF in a way, but you have all these additions, made all these additional steps to it.
That's really interesting. I honestly never heard of that technique. It's really interesting. And I can imagine, as you said, that it takes some time to master that skill. So what you were explaining on all these steps, once you have everything fine-tuned and optimized, I guess that the hope is that the outcomes, the clinical outcomes are going to be standardized and optimized. Do you have any clinical data so far?
Yeah, so we have published one paper. We've presented 2 or 3 abstracts in meetings, both ASHA and ASRM. And the idea is to publish, we did a first series, which we called the proof of concept study. And that stopped around May, June last year. So it's only a year ago. We do vitrification in all the embryos, manual or automated, okay? Focus there has been mostly manual. And that means that you have a delay of 3, 4, 5 months before the patient returns. So it's been a long process of about a year. We have published one paper on the proof of concept study on remote ICSI. Was published about a month ago in Reproductive Biomedicine Online with Gerardo Mendizabal, the chief engineer, as first author. And so that's the first one on the first baby born from our automated ICSI process. It involves all the sperm steps. It involves all the egg steps. There's not a single step left out on the microscopic version of it. But of course, in ICSI, as an embryologist, you notice you also have to add the eggs, right? You have to add sperm to a PVP droplet, and you have to take the eggs out and maybe wash them or not, depending on your preference. So there's all the other typical embryology steps that I don't call necessarily in this context microscopic, but they're still done on the loop, a microscope, compound scope. So that wasn't included in our next line, the ORA line, where we build all the stations from scratch. Their ICSI will be ultimately involving all these other steps as well. But yeah, we have been able to do all the steps of ICSI automatically. In the proof of concept study, we're writing several other papers about that study. And then the ORA line, which is developing tools in their own stations. It's actually 5 stations next to each other. Those can be miniaturized or made smaller later, but now we've given ourselves space. That data will be collected in our first sibling oocyte study. You're looking at papers coming out in 2026. It's always like that. Reviewers don't accept if you don't have babies at the end. They just don't accept it, even if it's a concept study. And so very rarely do they do it. You can't take that risk as a researcher. So, you have to wait quite a bit of time after the last patients are done. So you're looking at 2026 publications there. But this year will be the proof of concept study, which I think would be nice to.
I think a lot of people is really looking forward to seeing that data, and there's a lot of interest in the platform and just the technology that you're developing. And I think it's important to touch on the topic on how the role of embryologists is going to evolve in our field, given that we have this technology coming where they can potentially, in the future and years from now, automatize almost all of the steps that they're doing in the lab. How do you see evolving the position of an embryologist?
Yeah. There's a doctor in Vietnam, I think it's Professor Hong Hanoi. She just was at the IVF meeting, and I didn't attend her lecture, but I heard that she described it like AI, and this was about AI, it wasn't about automation necessarily. But she described it as, well, you're not going to lose your job, but you're going to lose your job if you don't use AI. And I like that. I think it's like in the early days when freezing came about, in the first 3, 4 years, 5 years of routine IVF in the 80s, freezing didn't exist yet. It hadn't really been tried in human IVF. And once that was done, it still was only a few laboratories that could do that for a few years. But then it became routine. Each clinic had to add it, because otherwise the alternative is to throw embryos away, which was totally unacceptable, of course. And so it was surprising to see that some embryologists just didn't want it. They were reluctant. It was just terrifying for them. You probably don't understand that, but you need to understand freezing what had not been done. It was a pioneering step forward. As the same for microsurgical fertilization 10 years after that. People were very excited about ICSI and very excited about the methods before, less excited about the methods before because they didn't work that well as ICSI. That's why ICSI completely took over and obliterated everything before it. But there was apprehension, and embryologists that didn't take on freezing and later didn't take on microsurgical fertilization, they were marginalized. At the end, they were marginalized in the different laboratories. They may have had important administrative functions. They may have been only doing egg retrievals and only doing sperm preps, but at the end, they didn't develop. They didn't evolve in their profession. And so that's what you're going to see here. If you think you're an embryologist, you don't want to deal with AIs, it's over your head and you're scared of it. I understand, but then you're missing out because these are tools that help you and assist you. AI cannot be done without an embryologist, and AI and automation for the longest time cannot be done without embryologists. Embryologists have to be in charge, but not only be in charge, check all the different aspects of it. And so automation of sperm prep, autonomy will be that you have a lab that processes sperm, every aspect of it. I don't see that happen, every aspect of it. I don't see that happen for a long time. I don't want to put a year on it, but it's going to take a long time. It's going to take a long time to do things like tridification in an truly autonomous fashion. This needs to be all supervised by embryologists step by step. I think that the future of it, as an embryologist, embrace it is my advice. Embrace it. Yeah, it's terrifying. I'll be terrified too if I was still an embryologist. But embrace it because you're going to like it. At the end, you're going to like it. Maybe not the first 2 months, no, maybe not even the first year, but you're going to like it because the tools will get better and better. You have to grow with it. It's not going to be one invention. It's going to be 1,000 inventions. That's what this field is promising. Automation in AI is 1,000 inventions. Whereas freezing in the beginning was just slow freezing with a couple of variations and later vitrification, but it's much more concise. It's a much smaller, in a way, if you look at it from the amount of effort in engineering and invention, this new way is massive in comparison. Grow with it. So as an embryologist, I think this is the best time to become an embryologist or to be an embryologist. This is the best time. Embryologists saying, should we advise our friends to become embryologists? My answer is yes. This is the best time. You're going to have a really good time. And the things that you weren't that good at as an embryologist, that's going to be part of the past. You're not going to have degeneration from denudation at the end. In the beginning, these machines may not work perfectly, in your opinion. You may even feel that it is never going to replace an embryologist. At the end, it will be so much better than what embryologists can achieve. It's unavoidable, but the whole process is, I think, a long time in the future. This is the best time to become an embryologist because at the end, you'll become both an engineer and an embryologist. I think it's wonderful.
I can see how for some people, they could perceive AI and automation as something really exciting that is going to improve the way they work. I can see how for other people it might feel like a threat, especially if they don't want to jump on board of it and it feels unknown and new and still not fully developed. But I also wanted to bring up that there's been a couple comments that I know that didn't please most of the people in the field, and this may be outliers, but there were a couple of comments on LinkedIn that were describing how automation is going to improve the outcomes. But in explaining that, they made a few comments of how an embryologist could be, and I'm going to read this from the quote, hungry, fatigued, distracted, or even hangover. And I think that people felt a little bit disrespected by portraying embryologists in that light.
Yeah, and I can understand that because particularly the last one, fatigue, yes, embryologists will acknowledge that. It's a very stressful job, as I said before. It's a very stressful, intense job. And so, yeah, you're going to be tired most of the time, maybe at the end of the day, but often could be in the morning because you're just tired from the 7 days that you did nonstop embryology before, or even 3 days. There's a reason I did it only for 20 years or so. I think it's a very difficult job to do day in, day out. But this hangover comment, that is outrageous. And to generalize is always difficult, right? To say embryologists are always going to be tired or they're stressed out. Yeah, sometimes you are. Most of the time, I think you're with it. And you're solid. You have the command to do it. You have the ability to do this properly and have a stable outcome. So these are generalized comments. It's very social media. My generation has a problem with social media. This is not how we were raised. We were not raised to make ourselves look at as a king or a prince. And that's the norm now. So let's not take this face value. This is somebody saying something, and some people have agreed that maybe this is a common comment that you hear more often, but this is generalized. And therefore I take it with a grain. Yeah. The comment on hangover is outrageous. That is outrageous. Has an embryologist ever had a hangover while doing embryology? I can't bet my money on it. I think it's extremely rare, but Certainly pilots have. So I think if you look at it from that perspective, probably a lot of professionals, occasionally in a lot of professions, this happen, but it's outlier. It's outlier stuff. I think it's ridiculous. Let's not generalize.
I appreciate getting your thoughts on that. I felt it was important to touch on this
Yeah.
because I agreed with people feeling disrespected by these comments, and I
So I can
appreciate
imagine.
you sharing your thoughts on this.
Yeah.
So beyond automation, there's AI that is definitely involved in automating the IVF processes, but there's also AI, and you mentioned before that there's over 30 startups right now in IVF developing AI tools to help us. What are your thoughts on those?
Thank you for that question. So my thoughts are I'm a little fearful for the people working in in IVF AI companies. Now, embryologists are fearful of losing their job on AI. I think it's really the people who work for the AI companies that are at risk of losing their job. That's simply based on statistics, okay? If you look at what we call Silicon Valley in the United States, I think a lot of people in the world refer to the world of startups as Silicon Valley. If you look at statistics in Silicon Valley, more than 90% of startups fail. More than 90% of startups fail. It's a daunting fact. Okay. And so is that different for startups in medicine? That's the first question to ask. I haven't found any evidence that it isn't. So 90% of medical startups fail. And there are different types of them. There are the service related ones that you're setting up a new IVF clinic network. That's the startup. You could get funding for that. It's actually very likely if you have a good business plan that you get funding. And so though the likelihood of that failing is very low, actually. It's much better than the failure rate of 90%. It's much better. It's a failure rate that is well below 50%. But if you go to technology, like developing a new way of increasing the fertilization rate biochemically, or working with trying to develop more eggs from somatic cells or from precursor cells, stem cells, I think then you're in a high risk area. So with robotics and automation and AI, you're also in a high risk area where the failure rate is going to be, I think, well over 80%, 70%. And so most of those companies are going to disappear. And there will be some that will make it, 2, 3. And there will be some consolidation. What I always hope in startup technology companies is if it's not business related and it's not about business plan for a network, what I always hope that the concepts, the IP will survive. So, and it usually does, as long as you share in terms of publication or you share your technology, even in patents, you're sharing your technology if you patent because it's publicly available in great detail. More actually than the details that are provided for peer review papers, publications. So yeah, share as a patent or share it as a publication, then the technology will survive. And even if the company doesn't make it, a good part of that IP will then live on. So I think that is important. There's a long answer to your question. Sorry.
No, it's a really good answer. And it just makes me think because it has a good overview of our field, plus you're invested in working with AI automation. So you have great combinations to potentially answer my next question, which is what do you think are the key ingredients to become one of those top 2, 3 startups that are going to break through in AI?
Yeah, that's more, looking at the future and crystal ball. And I don't have that ability to give you a good answer, I think. It will require a good business plan and be flexible enough to change that plan if needed. It will require talent. In the case of automation and AI, I think that talent is, of course, embryologists like you and others who have to be involved and criticize or try things out. So it's not only the people, the embryologists that are involved in actually developing the technology, but embryologists involved in trying the technology for the first time clinically. I think it's extremely important to give that feedback and give it in a massive way. And one of the criticism I have, and this has nothing to do with automation and AI, although the same thing will happen there, but it has to do with my experience of developing other areas in IVF, particularly PGTA, where I must say, Santiago Munar and I, and people working for us or with us, struggled for literally 1.5 decades to get data back from clinics. They wanted it. They don't want to pay for it because this was all experimental. and then they'll give you the data back. And this is rampant through our field. It's actually incredible to see how few of the companies where you provide an experimental method just for testing, and you can't get the data. And by that, just a few data points that are important. KPIs, it's not that much. It's fertilization rate, it's usable blastocysts, and getting the data back about pregnancies and birth outcomes. It really is not that much. It's little effort in return for something that's important. And so this has been, if you ask me what's about to be my biggest struggle in startups, that would be my answer. That's the biggest struggle, is that we don't get the data back. We do not get the data back. And I see that the same as other AI companies. So in short, answering your questions about which ones are going to make it, the ones that are going to get the data back, those are going to make it.
I can definitely see the struggle from both sides in this topic, because from a clinic perspective, they're so busy that even if it's a few data points, it's so hard for them to collect them, to get the time to do it. But then from the business, and their criticism would be, you're trying to sell me a technology and you don't have a solid study, but you're trying to put together the study, you're struggling to get back the data. So it's hard.
It's a vicious circle. Yeah, it's a vicious circle. Are we innocent on the developer side? No, but a lot of these pioneering clinics, and I always appreciate them, the pioneering clinics expect to get everything for free, and all we need in return is data. And that's happening right now with AI. These companies are often offering things for free. Some do not. We're offering it for either very little or for free. And so give them the data back. It's important. And I know that embryologists are busy, and I know clinicians are busy, and nurses are busy, because often the data also has to come from nurses. But it's essential.
And I wanted to ask you something that's going to sound romantic from a scientific perspective, but I feel we're so invested in selecting the best embryo, the best sperm, doing AI to optimize the process, automation to standardize. Have we forgotten to do basic research and try to solve other problems to improve the sources, improve the eggs, improve the sperm from the beginning, so we can improve the quality of the embryos and the quantity of the embryos? What are your thoughts on that?
Yeah, there are more than AI and automation buckets to research. There are 4 or 5 buckets, areas where research will always be important. And so the culture system, is it being sorted out? No, it's not being sorted out. And so there's always work to be done on the culture system. I am a little worried that with the aging of the pioneers in the embryo culture system, fertilization culture system, I'm a little worried that those typical molecular biologists, we used to call them biochemists or metabolic experts, that those few that have made this field big, that they're either have retired or are about to retire in the next 10, 50 years. So I'm a little worried about that, but it's central. If we know that IVF has become better over the years, not in every country, but in countries that keep statistics like in the United States, it's very obvious. And if you look at the British HFEA statistics, annual report that comes out, failure number 1 is how has the possibility of an embryo implanting and making it into a baby or developing it into a baby, how has that changed over the years? And they look at it for maternal age groups, and you see all these curves go up over time, starting with a time that's 20 years ago or longer than that. You see it goes up every year. So you need a large database for that. So you need to combine a lot of clinics to get that kind of data out. So we know we've gotten better. What's the main reason for that? There are many reasons, but I think one of them is really the embryo culture system. The embryo culture system we had at the round of Louise Brown, and in the early 80s, when, around 1980, 81, the first clinics were opening around the world. By 81, it was maybe 5 or 10 clinics, very small number. But that culture system, it was lousy in compared to what it is nowadays. It was for cell tissue culture specialists. And even for them, it was not a good culture system. So the understanding the culture system, the differences, the various physical, the importance of osmolarity and pH and temperature, to understand the basics of that has taken decades. And there is still improvement possible there. There really is. So that should not disappear. but I'm a little fearful that it may become commonplace. This is it. And now it's just the manufacturing process can be perfected and all that. But I think there are still major questions to be answered. Science of that. So I think that's the long answer to your question. That's the most important.
It's a great answer.
That's the most
Yes.
important. Of course, everybody's looking at AI and automation to give you the answers, but I think automation will, and AI too, will give you standardization. You've mentioned this a few times, standardization. An advantage of standardization is that once you do the randomized clinical trial in a standardized process, where you have eliminated a lot of the variations that you could not eliminate before, I think then those trials become more valuable. If you look at randomized clinical trials done in the human IVF lab, there's very few things we've learned from it. Very few things. It's terrifying, really. One thing we've learned, and it actually took a while, was that you need to culture under reduced oxygen. That's one of the things we've learned. But experimental embryologists, mammalian experimental embryologists, have known that since the 1950s. So we're talking 70 years later. And that was only affirmed in evidence-based IVF medicine something like 10 years ago. I think the part of the problem is of randomized clinical trials in the IVF lab is this tremendous variation. So you need to do thousands and thousands of cases, translating into tens of thousands of oocyte embryos, preferably, to actually make sense of it. You often see trends, and they say, Liz, if you increase the numbers, you're probably going to find a difference. That's the problem. And maybe we don't have to increase the numbers if we have standardization.
I was wondering why, in my opinion, have there been such a disconnect between basic research and translational research, or what we apply in IVF in medicine? Because there are so many things that we could have already found in research done in animals or bovine industry, for example, that we are not applying to humans. Do you feel the same way, or maybe a bit negative?
I think there's a reluctance. It's on a governmental level, and research support in most countries in medicine is very poor in most countries, either non-existent or poor. And so if you look at the United States, the NIH, and I don't count in the new administration because they're on a different level. So I don't want to go into that. But NIH has been the largest funding agency for medical technology, and at a level of $60-70 billion a year. That's huge. It's larger than all of European countries combined. I think it's actually larger than all of the countries combined. And so if you look at that, how much money has been spent by NIH, the National Institute of Health, on human embryo research? I can tell you, I know exactly the number. It's zero. That's the number. Zero. Anything between egg retrieval and embryo transfer has never been supported by the American government. It was only under Bill Clinton that in his first months, the second decree he signed, which was in January 1992, was that we should open it up for human embryo research, the NIH budget. And that was killed very quickly by Republicans. And that's been the trend. So since 1979, since it was clear that human IVF was a possibility, by presidential decree first, paying out of the pockets of taxpayers for that type of research was prohibited. And then later it became law in 1996. It became a law that you couldn't get tax monies for supporting human embryo research. Is there any other field in medicine that has been handled this way? No. And even in reproductive medicine, you can get support, but just don't mention that you're doing IVF. You can't have an IVF laboratory and have some observation, even in a social way or in a demographic epidemiology way that had nothing to do with the IVF lab, but you're using that data, you cannot get money. So isn't that an amazing thing? And that's how it's been in most countries. I think with the exception, really, truly, the only country where it's been an exception is Belgium. And other than that, occasionally Spain has been an exception. Occasionally the UK has been an exception. But it's only Belgium that has been very firm that there must be possibility to get federal grant money for human embryo research. That means between egg collection and including egg collection and embryo transfer, including embryo transfer. This is the reason why things go so slow. All these agencies just don't support this. There's always somebody who has ethical,
Yeah.
and it's all about ethics and objections to what this is, not understanding the fundamental importance of what's being done in human embryo research. We could be finding out it's about the origin of life. Okay, so not supporting that is astounding. But it's been like this since 1979.
It's just shocking to think that in so many years that changed. The regulation and the support to research in our field has changed so little.
No. So how can you get money to do these studies? And you call it translational research. And the problem with translational research, if we look at the mouse model or the bovine model, there you can get monies to between getting eggs out and transferring embryos to animal systems. Of course, you can get support. Not that easy, but you can get support. That's not necessarily translational research because these are models. But I always have on my list of papers to write mouse or man, because in our field, has the mouse model really been helpful? It has sometimes, but not all the time. And actually has done detriment too. And so it's just a model. It's not really translational. We need to do it. You need to test it in mouse. All of our culture media is all the time tested in mouse model. Zygote test, or we do IVF, we don't do zygote. Okay. We have these models to test for toxicity of culture media. And it has to do with the manufacturing process, not necessarily with the formulation. Because the mouse model accepts most formulations without discrimination. And so it's helpful. That's all we have, apparently, although there may be changes in that area. I hope there will be. But it's not a translation. And that's why things go so slowly. We need to test, we need investigation on human IVF and human embryo development. That's where the real progress would be, and we don't have that support.
For a closing remark, if you were to describe using 3 words or just a few words the future of our field, what would those be?
I think we have outlined already what it is. It's genetic research, obviously. I think we're still on the tip of the iceberg there. It's wonderful to see how much the PGT community, the researchers have figured out things over the years. It's great to see that. I think that needs to be explored further, definitely. So that's one area that will be ongoing. Embryo culture system, you pointed that out. Embryo culture system is something we need to keep working. And then, of course, there's AI, just AI. And then there's AI combination, particularly computer vision in combination with automation. Those are the 4 areas. I personally think that the real breakthrough that will change our field has to do with gamete engineering. The fact that we're still doing follicular stimulation now 50 years after, more than 50 years after Louise Brown, 47 years, but soon 50 years after Louise Brown, I think is astounding. That really limits the application. It's labor intense, very labor intense. It's expensive, and it is hard for female patients. And I think that needs to change. So gamete engineering is an effort that I think we all should be supporting more than we do. And then I think that is the overview where everything is placed. So it's genetics, culture, gamete engineering, AI, and automation, those 5.
Before we end our conversation, I wanted to say something. I don't know if someone is going to stay for the whole episode, but if they do, I just wanted to say something about you, Dr. Cohen, because sometimes we can read papers, we can read your bio in a website, or we can see you making these interviews, but it's hard to tell about the person. And just sharing an anecdote, this is the 3rd time that we are recording this interview. It takes a lot of time and patience to do, especially for someone that has the smallest, tiniest social media platform. I just started, but I'm doing this with a lot of love. And I think that tells a lot about you, about your dedication, and that explains why you have the career that you have. So wholeheartedly, thank you for your time and being part of this. I have learned a lot in this conversation with you.
Thank you. And thank you for your excellent questions and your patience. This is the 3rd time we're doing it. I think in both cases, both previous cases, it was all my fault. So one of the reasons But it's a pleasure talking to you, and I hope you will be successful in this effort. It's important to do. Communications is one of the blessings of our current age, and I think that we need to pursue this intensely. I was involved in one of the founders of the International IVF Initiative, and so I've always been in favor of working on platforms like this. So I hope you'll be successful. Been a pleasure talking to you.
Thank you, Jax.